baby?
It's Scott.
As a baby.
He'll grow. Time travel! I see this as an absolute win.
Be strong.
All right, today we're going to talk about mitosis. Let's talk about some fun facts. Mitosis overall, it occurs in somatic cells. These are our body cells, which is basically every cell in your entire body other than those gametes that we're going to learn about. Remember, our gametes are our sex cells. So in males, it is sperm, and in females, it's egg. Mitosis makes identical cells, since we want, if we have a toe cell that needs to be regrown, it needs to be the same. And it goes through a process called PMAT. We're going to learn about it. So before we move on, let's review the cell cycle. Remember the cell cycle, if we start here at birth, overall, the first thing your cell is going to do is just going to grow. It's going to get bigger, and then one day it's going to say, I'm too big, so I need to make a plan so I can divide. So then it goes into something called the S phase, and this is where we copy our DNA. And as soon as we copy our DNA, we do that DNA replication, we need to make sure that we didn't mess anything up. So we're going to go through G2 and we're going to revise it. So now let's talk about the actual dividing phase. The phase that we talk about in somatic cells, the M phase stands for mitosis. It makes up 10% of the cell cycle, so it's not a lot. Remember, interphase is the other three, G1, S, and G2, and it's 90%. So this is only a short segment in the life cycle of a cell. So let's go through the 4 steps that make up mitosis. Step number 1 is prophase. We start off with prophase. It just looks like a dot, basically, when you look at a cell underneath a microscope. It's not that fancy. The only things that happen in prophase is we need to get the DNA out of the nucleus, so the nuclear membrane breaks down. And then once we break down the nuclear membrane, we're also going to make something called spindle fibers. And these are kind of like the fishing rods that are going to come in and move our chromosomes apart. And if you don't remember chromosomes, please look at the video associated with that. Step number 2 is metaphase. Meta means middle. So our sister chromatids are going to move to the center of the cell, or the cell plate, and they're going to line up. In mitosis, it's a single file line of X's, if we can say that. The spindle fibers will then attach to the centromere, or the center of those chromatids, and it will lock in so they can be pulled apart. What's going to pull them apart? Those little red things at either end or either pole of the cell, known as centrioles. Those are like the fishing reel, and they're going to pull them and separate them into 2 pieces. Step number 3 is where we actually separate their sister chromatids. Ana means away. So the sister chromatids are going to move away from each other, and they're going to move towards the poles in the cell. So if we look at our cell, it's starting to stretch as if you were stretching, a ball apart, and you see those sister chromatids flying away, being pulled by the centrioles that are attached to the spindle fibers. All right, so let's look at phase number 4. This is telophase. Now our cell's starting to be pinched like a balloon. If you imagine you were pinching a balloon in the middle. The only problem is we can't separate it yet because our DNA, those sister chromatids, they've been pulled apart, but they're still vulnerable to being sucked out of the cell. So what do we put them in to keep them safe? Well, we're going to put them back in a nucleus. We need to reform that nuclear membrane. When we
Say again, please?
Houston, we have a problem.
Let's think about this. We have a nucleus. We have DNA. We need to protect the DNA inside the nucleus. So I think about it, if you've ever seen a movie with like a spaceship, they have the airlock. Well, right now in telophase, the airlock has not been reformed yet. So if we open the door to the spaceship, everything inside is going to be sucked out. So the point of telophase is we need to close the airlock. That way when we break the cytoplasm in the next step, all of the contents of the cell will not be released. Specifically, the most important content, the set of instructions that is going to build the entire cell, the DNA. When we do that, we have something called a cleavage furrow that forms where we pinch that cell where the cell membrane is getting ready to separate. And then the last step, technically it's not part of PMAT, depending on how old you see them talking about mitosis. In recent years, we've separated it into its own step. It's called cytokinesis. Cyto means cell, kinesis means apart. So now I have 2 cells that are apart. It splits, literally the cell membrane or the plasma membrane splits in half, and we have 2 new identical cells with DNA contained inside their nucleus, and we call them identical daughter cells. It's more of a process than a step. It's just you're cutting the cell in half. Overall, mitosis is 10% of a cell cycle for a somatic cell. The 90% left over is for interphase. It is made up of 4 steps, and PMAT, prophase, metaphase, anaphase, telophase, cytokinesis is our last step. We ended up with 2 identical cells, each with 46 chromosomes that are diploid. Next, we'll talk about meiosis.